Hi everybody, we're going to do a walkthrough of excitation contraction coupling. Now this is different from the PowerPoint narrations that I've provided for you or through other animations. I have a completely blank screen here and I would like for you to pull out a few blank pieces of paper and work through this with me. Now in your course pack, this information is pages 83 to 85. So first of all, excitation contraction coupling is two separate events. Excitation is the excitement or the stimulation of a muscle cell. We do this by using a nerve cell telling that muscle cell that it's time to contract. The contraction event is a separate event. So when we're talking about excitation contraction coupling, keep in mind there's actually two separate events here to take place. So I'd like to start off by talking about excitation first. So I'm going to draw here an axon. And of course, this is an axon terminal. And axon terminals are just branch endings of our nerve cells. And axon terminals sit really close to nearby muscle cells. So I'm going to draw here for us a sarcolemma of a muscle cell. And these are protein channels here. They're not just any old protein channels. They are, in fact, sodium channels. And inside here would be the sarcoplasm. That is the cytoplasm for this muscle cell. And then down here near the bottom, this would be the other portion of that plasma membrane known as a sarcolemma. So first thing you notice is there is a bit of space between this axon terminal, that's of course part of a nerve cell, and the nearby muscle cell. Muscle cell, of course, being this one down here. Do you know the name of that space in between the two? That space is called a synaptic cleft. And what we're going to discuss occurs in that space. Sometimes it's just simply called a synapse. Okay, so when the axon terminal wants to stimulate the nearby muscle cell, you see we've got a problem. That axon terminal can't tap the nearby muscle cell and say, hey, time for you to do a contraction event. So instead what we have to do is use a chemical messenger to travel across that synaptic cleft and notify that muscle cell that now's the time to contract. So we're going to start with an action potential, which is just fancy language for a nerve impulse that travels down the length of that axon terminal. When that nerve impulse arrives here in the axon terminal, there are special channels here called calcium channels. And they will open. Okay, so nerve impulse travels to the axon terminal. That causes these calcium channels to open, and calcium comes into this axon terminal. Now you got to have calcium in that axon terminal for the next step to happen. The next step involves these little vesicles here, which are full of neurotransmitter, to move closer to the end of this axon terminal and rupture like a blister. Now, the particular neurotransmitter we're going to discuss is called acetylcholine. So acetylcholine is the messenger that travels across this very tiny synaptic cleft and over to this muscle cell. ACH will then travel here and bind to a receptor site on that sodium channel. Okay, let's do it again from the top. When a nerve impulse travels down the axon terminal, that stimulates the opening of calcium channels. Calcium channels open, allowing calcium to flow from the interstitial fluids into the axon terminal. Once inside, calcium allows for these vesicles, right here, to move toward the end of the axon terminal. These vesicles hold a neurotransmitter called acetylcholine. They rupture like a blister, and acetylcholine travels across the synaptic cleft and binds to a receptor on this sodium channel sitting on a nearby, muscle cell. All right, so these are the steps of excitation. We're getting there. We're almost to the point of exciting this muscle cell. Once acetylcholine binds to these sodium channels will now open. So it's like putting a lock, key in a lock. Once you turn the key, that lock opens, the doorway opens. That means that you've got to have ACH sitting there to open up this sodium channel. Now that the sodium channel is open, sodium that's sitting out here in this interstitial fluid will travel into this muscle cell. So sodium flows from an area of high concentration outside the muscle into the interior of the muscle. And you'll notice there's a lot of sodium coming in here. And in fact, there's so much sodium, we turn the environment inside your muscle cell from a negative state to a positive state. That is excitation. So you brought so many sodiums in that you have now excited your muscle cell. So think of it this way. Think if you're having, the party of the year, you have kept the doors locked until the very last minute. And at the last moment, you fling open the doors and everyone floods in. That's what happens here with these sodium channels. Sodium channels open, sodiums flood in, and all of a sudden you have a wave of excitement inside that muscle cell. So it turns that environment inside your muscle from negative to positive. Now, we just completed excitation. Let's do it one last time from the top. First, a nerve impulse travels down the axon terminal. That nerve impulse is a wave of excitement. It causes calcium channels to open. When calcium channels open, calcium floods into the cell, and of course that axon terminal now prepares the release of these vesicles full of neurotransmitter. Neurotransmitter, ACH, travels across the synaptic cleft and binds to a sodium channel. Those sodium channels now open, and sodium now floods into your cell. That wave of sodium brings with it a positive charge, causing a wave of excitement now inside your muscle cell. Nearby, sodium channels will also open, and this wave of excitement will travel down the length of your muscle cell, which is what we call an action potential. Okay, so now we have completed excitation, and that should take us to about the bottom of page 83. Now, before we move on to contraction, let me mention that as long as those sodium channels sit open, we're going to see sodium rushing into the muscle cell, and a wave of excitement will continue. In other words, the message will continue to be, hey, muscle cell, contract. So if you want to close those sodium channels, you have to get rid of that ACH. And the way you do that is by using an enzyme that recycles it called ACHE. Acetylcholinesterase, or ACHE, takes the pieces of acetylcholine and recycles them back up here into that axon terminal. I put it to you this way. If you don't recycle acetylcholine, you will ache, ACHE, because if you keep those sodium channels open and your muscle stays excited, your muscle will stay contracted. That can make you ache, right? So acetylcholinesterase is an enzyme. It helps to recycle acetylcholine back up into that axon terminal. So it puts the muscle cell back to rest, which is critical. You don't want your muscles to sit in a contracted state for on and on. Okay, I hope that helps. That takes you down to the bottom of page 83. There's a picture in the top of 84 that's more or less what we've looked at here. Let's now continue on and talk about contraction. I'm going to erase this picture and we'll start with a different one. Okay, so let's talk about contraction now. What I'd like to do is say that we're inside of a muscle cell. So I'm going to draw our muscle cell. The plasma membrane we know is called the sarcolemma. And we know that inside that muscle cell, we have these long, skinny organelles called myofibrils. Okay, so the myofibril is an organelle inside your muscle cell, and these myofibrils are wrapped by like a lacy sleeve. Okay, and we're going to call that lacy sleeve the sarcoplasmic reticulum, or just call it SR for short. Okay, so let me just come over here and say that we have this T tubule. So when that wave of excitement comes down the length of your muscle cell, here's your muscle cell, those waves of excitement open up these sodium channels, and sodium rushes in deep into these T tubules. So we're getting sodium rushing in all along the length of this muscle cell. These T tubules dive in deep and carry the nerve impulse or action potential right to where these myofibrils sit. So when this wave of excitement comes, these sodium ions carry a positive charge. And that positive charge causes this sarcoplasmic reticulum to release calcium ions. Okay, so calcium is released once again. Second time we've included it in our conversation. And now that calcium goes into the sarcoplasm. And of course, sarcoplasm is fancy language for the cytoplasm of the muscle cell. So where we really need it to go is to head to these myofibrils, these organelles here that are the contractile units of our muscle cell. So now I'm going to take you into the details of the myofibril. So let's clear this image. And let's talk about a set of proteins here attached to these Z lines. And of course, they make up that myofibril. So what kinds of proteins are anchored to these Z lines? Are they thin filaments or are they thick filaments? Well, these Z lines are the anchoring point for our thin filaments. And of course, thin filaments are made up of 3 proteins: actin, do you remember the other 2? Troponin and tropomyosin. Okay, I jokingly have called them AT and T. Okay, so they are anchored to a membrane called a Z line or a Z disc. Now there are also thick filaments here, and they are composed of a different protein known as myosin. Okay, so myosin are thick filaments. And of course, they have these little bumps on them. They are called myosin heads. So we'll just label one of those as well. All right, so let's draw a few more of those thick filaments in, put their little bumpy myosin heads on there. One more setup here. Okay, so we are now ready to talk about some banding patterns that you may see here. The myofibril, of course, has light bands and dark bands, and the light bands are the ones that are centered around these Z lines. They, of course, contain no thick filaments. If we circle the next region here, we are looking at the A band, which of course contains those thick filaments. This specialized area right through here would be our H zone because there are no thin filaments in the H zone. So as soon as this muscle cell receives some calcium, of course, the calcium is a very important ingredient because it allows for us to release a rope-like tropomyosin from the binding sites on those actin filaments. As soon as those binding sites are available, these little myosin heads can reach up and form a cross bridge. When that cross bridge forms, it then can pivot and move those thin filaments, okay? And so we say that movement is called a power stroke. So when we move these thin filaments, they are being pulled or slid. Keep in mind, the name of this concept of contraction is called a sliding filament theory. So as these heads reach up and bind to these thin filaments, they are forming cross bridges, they pivot, forming power strokes. So that only moves it so far. So how do you repeat the process so you can continue sliding those thin filaments? Well, you give that head some ATP, and then of course it releases and then it reattaches and pulls again. So eventually what you end up having is the entire set of Z lines moving toward the center of this sarcomere. We know a sarcomere is the distance from Z line to Z line. So as these heads attach, release with ATP, reattach, pull and slide, we end up causing our sarcomere to shorten. And so the sarcomere is the only thing about this myofibril setup that actually shortens during muscle contraction. The thin filaments nor thick filaments actually shorten. We do lose the I bands, and what else do we lose? We lose that H zone because those thin filaments invade that area. So ultimately, we end up with muscle shortening thanks to the sliding of thin filaments past thick ones. All right, so that should take you over to page 85. Now, in order to make this muscle relax, we need to restore the calcium here back to the SR. We also need to have acetylcholinesterase. remove acetylcholine and recycle it back to those axon terminals. So eventually these muscles will sit back to their relaxed state. The sarcomere will lengthen once again and return to its relaxed status, not a contracted status. So I hope these drawings and working through this with me has helped. Please reach out if you have any questions. This does cover pages 83, 84, and the top of 85.